Welcome, ladies and gentlemen, to our online lecture on the specific steps of cellular respiration. As we work our way through this lecture, your focus is on identifying the inputs and outputs of cellular respiration and describing specifically where things are used and produced. So while there's an overall reaction for cell respiration, not every part gets used in every stage. So one of the things that I will expect you to know is specifically where each part plays a role in the system. So for cellular respiration, there are three parts. Glycolysis is a precursor. This process is done by all living things. So bacterium, eukaryotic cells, everything can do glycolysis. The next part of the process is called the Krebs cycle, and we will only proceed into this if oxygen is present in the system. The reason why is that the parts that are created from the Krebs cycle need to go into the third step called the electron transport chain. So if oxygen is not present, which happens to be used in the electron transport chain, or ETC, the cells will not do the Krebs cycle because there will be nowhere for those products to go. You can see the overall diagram below, but we will be talking about each step specifically. Now, the word glycolysis literally means sugar splitting. So glyco is sugar. And lysis to split. If you recall, when we were talking about cells responding to hypotonic environments, we said for a cell to burst like that, it is lysing. So this initial step allows the particles to go into the Krebs cycle more easily. And with the right form of the molecules, it'll make it easier for the enzymes in that system to be able to make the correct products. Again, this happens in prokaryotes as well. It is not done in the mitochondria. So if it's not in the mitochondria, you might say, well, where does it happen? It happens in the cytoplasm, so in that fluid-filled space anywhere within the cell. I'm going to start with a glucose, and you can see that represented down in the lower left with the dark spheres. So throughout these diagrams, anytime you see a sphere like that, that's going to represent our carbons. And a big part of that is going to be tracking how many there are and where they go. You will see that in order to get this process going, the first step is to split the sugar in two, and to get 2 3-carbon pieces, that's going to take some energy to do so. So if I'm going to split that, the ATP are actually contributing the phosphates, and it's rearranging the molecules. So as I break the ATP and the ADP plus the phosphate, I also break the glucose and create those bonds with the phosphates on those smaller sugars. Then through a series of chemical rearrangements, we're going to capture some electrons. And as we rearrange things, we're going to end up with what we call pyruvic acid. So the electrons will be picked up by the electron carriers in the form of NADH. Not sure why the board keeps jumping back and forth on me. At the end of this process, in addition to the pyruvic acid and NADH, which are matter, I have some chemical energy in the form of ATP. While I make 4, only 2 are my output, because remember I had to spend 2 to get the process going. So while I create 4, there's only 2 more than what I originally started with. Now, an advantage of this process is you might say, man, I only get to make 2 at a time. That doesn't seem like very much. But the process can occur very quickly, so in even a millisecond, I can create thousands of ATP. The other benefit is that if oxygen is not available, there are still ways for cells to create this necessary ATP. Now the next step is going to now create a lot of electron carriers. We saw some coming out of glycolysis, but it was only a few. The main point is to capture electrons. They represent a source of energy. So if I have many of these high energy electrons, I can do something with that energy later on, which will be used in the electron transport chain. Now this is going to occur inside a mitochondrion. So when oxygen is present, we can proceed. The matrix is the middle. So those pyruvic acids are going to come out of glycolysis and get modified a little bit to acetyl-CoA. You'll see that in that process, I go from 3 carbons to 2 carbons. And that conversion from 3 carbons to 2 is where the one of the carbon dioxides comes off. So that is one of the outputs. It's the first time we've seen one of those parts of our chemical reaction. Then that 2 carbon molecule is going to go into the Krebs cycle, which involves a series of molecular rearrangements. So as the molecule gets modified, we lose some more CO2. We're going to create a little bit of ATP. And then we're going to generate many of those electron carriers. So it's a little bit out of view there, but you'll have 3 NADHs being produced with the 1 FADH2. So when you add together all of the NADH from the conversions, we're going to have 4 total going on for one turn, and 1 FADH2 is just another type of a similar molecule. The trick is we had 2 pyruvic acids coming from glycolysis. So that means we need to run the system 2 times. So for all of this input, you're going to double these numbers. So what are our totals so far? We produced 4 usable ATP, 6 if you count the ones that are offsetting the ones that went into glycolysis. We have 10 of those electron carriers in the form of NADH, and we have 2 FADH2. And all of those are now going to go into the electron transport chain. And remember, they are carrying those high energy electrons. Now the benefit of the electron transport chain is I can make many ATP at once. So compared to those measly 2 we get from the Krebs cycle or the glycolysis, I can make many times that. It's going to take advantage of those differences in concentration. So we're going to force a bunch of H+ ions into a space to create a difference in concentration. Since it's already in high concentration there, it's going to diffuse back through ATP synthase, which is a special enzyme that creates those molecules. So what does this all look like? Along the inner membrane, so remember in all of those folds, we're going to have the enzymes and the proteins involved in the process embedded in that membrane. The electrons start out as high energy, and think of falling as giving it up. If you started at the top of the hill, say on a sled, you would have high potential energy. Here in the winter, if you slid down that hill, you would convert that energy into movement or kinetic energy, and then you would have low potential energy at the bottom of the hill when you slide down. That's kind of what the electrons are like, is they have high potential energy to start, and as they move through these proteins and give up some energy to pump those H pluses, they're going to be low energy by the time they're done. So the proteins will pump H plus to the intermembrane space. That will collect many H plus in that space. When there's a lot in one place, remember diffusion will occur when things move from high concentration to low. That movement goes through ATP synthase. That movement of the H plus ions powers that enzyme. It's actually like a little motor. That part at the bottom will actually spin as a part of making the ATP. Now those electrons have to go somewhere. At the end, they can't just be hanging out in no man's land. So oxygen will combine with H plus ions and those electrons to create water. So when the oxygen is used, the reason why is oxygen is really greedy for electrons and it pulls really tightly on them. So as those electrons move through, it's going to pull most tightly at the end, and it's part of what helps keep things moving through the chain. And then that is where our water byproduct comes from as it combines with the O2 and the H plus ions in the solution. Now, all of those NADH and FADH2 electron carriers are going to give up those high energy electrons. And again, as they move, it's what was powering the pushing of those ions, that active transport, to where they already were. Again, the water will form when the H plus ions, oxygen, and electrons combine at the end of the chain. So what do we get when it's all said and done? We get 2 usable ATP from glycolysis. We make 2 ATP from the Krebs cycle. And the electron transport chain will produce about 32. Again, much more than what we get from the earlier steps. This gives us an overall total of about 36 ATP from one molecule of glucose. So let's kind of track where this comes through. You'll notice I've color-coded the materials in the reaction for you. So the carbon and the oxygen from the glucose, the byproducts of what's left over, would come out as the CO2. Remember that we lost those during the Krebs cycle. The H's then become part of those H plus ions that are around that will move with the oxygen and the electrons to form the water. So the blue parts go together and the red parts go together. The whole point of cellular respiration is to create ATP, because that ATP can be put back into ADP and phosphate and provide energy for other things. So if anyone says, what's the point of cellular respiration? The point is to make ATP.